Hey everybody, I'm shooting this video for those of you that either missed or did not turn in the red cabbage lab. The reason that we did this lab, or the purpose for it, was to show that we can determine whether something is an acid or a base by using something called an indicator. And the juice of the red cabbage is a great indicator that is one of the ways we can determine if something is an acid or a base. Now, before I get started with the lab itself, just a quick reminder of things that I need to see on the lab assignment. The first one being a written version of the procedure. And then you guys are going to draw two lines similar to the ones up here. In fact, exactly like the ones up here, starting at 0 and ending at 14. One of them you guys will be using to guess what the pH is of the following substances, and the second one is where you will plot the actual pH, or at least what you can guess based on what the indicator says. Now, the 7 different solutions that we will be working with can be found right over here. This is the list, and that should also appear on your lab report underneath your procedure. Now, the other things we need to know during the procedure are the 4 other pieces of equipment or materials that we've got to work with. The first, and maybe the most important of all of them, is going to be what we refer to as the red cabbage. Now, the red cabbage definitely does not look red. It looks purple, but it's referred to as a red cabbage, and it's got a special chemical in it called anthocyanin that actually allows it to change colors based on whether it's mixed with an acid or a base. Now, the other things that we're going to be using a mixer or a blender in order to get the juice out of the red cabbage. We have a glass of water because we need to dilute that juice into a more usable form. And we'll have to use a test tube as well in order to actually get this thing working. So, first thing I want you guys to do is take this group of 7 chemicals up here and see if, just based on what you already know about them, see if you can plot them on a line that looks kind of like this one right here. So this is what I'll refer to as the guess pH line. So what I want you guys to do, let's say you believe that vinegar shows up, I don't know, right about here. So this would be like a 6 or even a 5.5. You're going to put a little line right there and just write the word vinegar. And I want you to do that with all 7 of the things in that box. Now you can pause the video in order to do that. Once you guys are finished with that, I'm going to go ahead and describe what happens when I put each one of these 7 things into the juice from the red cabbage. Now, in order to get the red cabbage out, what you'll do, or the juice of the red cabbage out, I should say, what happens is you put the red cabbage, you take a couple leaves off, and you put it into the blender, you mix it in with the water, and you press the little button that says go. And as soon as it blends through, you get this nice kind of purple solution. And you can dump a couple, or take a dropper, in this case. You don't want to dump anything into a test tube. But take a dropper and just kind of take 2 drops and put them inside of your test tube until it fills up about 1/3 of the way. Once you've got that done, you've got your red cabbage indicator ready, and you can go ahead and start adding things to it drop by drop and seeing what the indicator tells you it is. So the first one that you're going to add in there is vinegar, and when you add vinegar in there, it becomes a very, very bright pink. So I'll go ahead and write that in. Bright has a T in it. Bright pink. Okay. So vinegar goes to a bright pink. Baking soda, on the other hand, turns into a light blue. Salt water also turns into a light blue, but it's not quite as light. It's kind of a medium blue. I'll describe it as that. A medium blue. Soapy dishwater also is kind of a purple solution. There's a little blue tint in it. It's kind of a purple blue. Hydrochloric acid, on the other hand, turns into almost a neon pink. It's even brighter than the vinegar. Ammonia turns into a light blue, almost a green. So that would be a light blue-green. Sorry if you can't read my handwriting very well. And then pure water actually stays as being purple. Doesn't even change colors. So we started, just kind of to recap, we started with a purple solution. When I added these 7 things in there, we get a color change ranging from hydrochloric acid at the most extreme pink end down to ammonia at the most extreme light blue or green end. And then pure water winds up somewhere in there. It didn't actually change the color of it at all. It's almost as if it's not an acid or a base, it's something else. So, what I want you guys to do, having been given that information, now you're going to go ahead and plot those on your second line, and this is what we refer to as the actual pH line. So, now that you've got the colors all arranged, you should be able to figure out what goes where on this line. And that will conclude your lab. Make sure you've got the procedure written down and your 2 lines, and I'll see you in class.